Okay, so let me ask you something. Where do plants get their food? Think about it. We eat plants or we eat animals that eat plants. But where do plants get their food? See, here's the thing. Plants actually make their own food. They synthesize it. This process is called photosynthesis. It's a physico-chemical process, which means it's both a physical and chemical process happening together. Plants use light energy to make organic compounds to build their own food. And because they can do this, we call plants autotrophs. Means what? Autotroph means self, troph means food. Self-feeders basically. Clear? Now everything else, you, me, animals, bacteria, we cannot make our own food. We depend on plants either directly or indirectly. So we're called heterotrophs. We eat other things for food. Now why should we study this? Why does it matter? Two reasons actually. First one, photosynthesis is the primary source of all food on earth. Not just some food, not just plant food, all food. Everything that eats depends ultimately on photosynthesis happening somewhere. Second reason, photosynthesis is responsible for releasing oxygen into our atmosphere. Think about that for a moment. Where does oxygen come from? It comes from plants doing photosynthesis. So all the oxygen you're breathing right now, that came from a plant somewhere. What would happen if there were no oxygen to breathe? You wouldn't be here, none of us would be. So photosynthesis is literally the basis of life on earth. The energy from sunlight captured by plants through photosynthesis, that's what keeps everything alive. That's why this entire chapter is about understanding how this amazing process works. Remember, you probably did this experiment yourself in earlier classes. You took a variegated leaf, one that's not all green, some white, some green parts, and you put it in light. Then you tested it for starch. What happened? The starch formed only in the green parts, the white parts nothing. So what does this tell us? Photosynthesis needs chlorophyll. That green pigment, that's essential. And photosynthesis needs light because you expose the leaf to light. So we know two requirements already, chlorophyll and light. But there's a third thing that's also needed. So another experiment you might have done. Here's how it works. You take a leaf, right? You cover half of it with a test tube that has KOH soaked cotton inside. KOH, potassium hydroxide, it absorbs carbon dioxide. So inside the tube, no CO2. Outside the tube, air is there, CO2 is present. You put the setup in light for some time, then you test both parts of the leaf for starch. What happens? The part outside the tube, the part exposed to air, it has starch. Positive test. The part inside the tube where CO2 was absorbed, no starch. Negative test. So what's the conclusion? Carbon dioxide is required for photosynthesis. Can you explain how this conclusion is drawn? Because if CO2 was not needed, both parts would make starch, no? But the part without CO2 doesn't make starch. So CO2 must be necessary. Let me take you back in time. It's 1770 and a scientist named Joseph Priestley is doing experiments. He takes a bell jar ash, that's a glass container, closed on all sides ash, and he puts a burning candle inside. What happens? The actual burns for a while and then it goes out. The flame extinguishes. He tries putting a mouse in a closed bell jar. Same thing happens, mouse. The mouse suffocates. So Priestley thinks burning candles damage the air. Breathing animals damage the air. They both use up something in the air. But then, and this is where it gets interesting. He puts a mint plant in that same bell jar. He keeps it in the light for some time. And guess what? The mouse stays alive. The candle keeps burning. The plant is actually restoring the air. So Priestley's hypothesis was simple but powerful. Plants restore to the air whatever breathing animals and burning candles remove. Can you imagine how Priestley conducted this experiment? He would need to rekindle the candle to test if it still burns. How would he do that without disturbing the setup inside the jar? Interesting question, no? Later in 1774, Priestley actually discovered oxygen, the gas that was being restored. So plants were putting oxygen back into the air. This was revolutionary. It showed that plants play a crucial role in keeping the atmosphere breathable. Without plants, without photosynthesis creating oxygen, all the oxygen would be used up by burning and breathing, and life would stop. So Priestley showed us that plants purify air, but he didn't show which part was important. Was it the darkness? Was it the setup? What was essential? That's where John Ingenhouse came in. He basically said, let me test the same setup, but in two conditions, darkness and sunlight. In bright sunlight, he observed small bubbles forming around the green parts of aquatic plants. What are these bubbles? In darkness, no bubbles. So sunlight is necessary. The green parts in sunlight are releasing oxygen. So now we know sunlight is essential for photosynthesis to happen. Then around 1854, Julius von Sachs added another important piece. He showed that plants are actually producing glucose when they're growing. Where this glucose? It's usually stored as starch. And where is this happening? He found that the green substance in plants, what we now call chlorophyll, moisturize, is located in special structures inside plant cells called chloroplasts. So, the chloroplast is the organelle where photosynthesis happens. The green parts of the plant contain these chloroplasts. And in these green parts, glucose is made and then stored as starch. So, now we're getting a clearer picture light, green pigment, and it produces glucose. Now, here's a clever experiment by T.W. Engelmann. He takes a prism, like Newton's prism that splits white light into rainbow colors. He splits light into its spectral components and he illuminates a green alga called Cladophora. Around this alga, he puts aerobic bacteria that need oxygen. Where would these bacteria go? They would go to the places where oxygen is being produced, where photosynthesis is happening. So Engelmann observed, where did the bacteria accumulate? They crowded mainly in the blue and red light regions. They avoided the green and yellow regions. So photosynthesis is most active in blue and red light. That's the first action spectrum, the wavelengths at which photosynthesis happens most actively. And notice, it matches roughly with the absorption spectra of chlorophyll, the pigments we have in plants. By the middle of the 1800s, scientists had figured out that plants use light to make carbohydrates from CO2 on and water. They wrote an equation. CO2 on plus H2 on with light makes H10O CO2 in plus a carbohydrate plus O2. Sounds good, no? But wait, there's a question nobody had answered yet. Where does the oxygen come from? Is it coming from the CO2 on that goes in, or is it coming from the water? This seems like a small question, but it's actually fundamental to understanding how photosynthesis works. A scientist named Cornelius van Niel studied photosynthesis in different organisms, purple bacteria and green bacteria. He showed that photosynthesis, at its heart, is about hydrogen. Here's the general formula. 2H2Na plus Ca plus CH2O with light makes 2A plus CH1O plus H2Na. Means what? H2Na is some compound that contains hydrogen. That hydrogen gets transferred, reduces the CH1 to carbohydrate. In green plants, what is H2Na? It's water. H2NO in purple and green sulfur bacteria. What's H2Na? It's hydrogen sulfide H2NS. Now here's the key insight. If H2NS is the hydrogen donor in bacteria, what's the oxidation product? Not oxygen downgrade, it's sulfur or sulfate. But in plants where water is the hydrogen donor, the oxidation product is oxygen. So the oxygen released in plants must come from water, not from carbon dioxide. This was proved later using radioisotopes. Again, put in title, what's H2Na? It's hydrogen sulfide. H2NS? Now here's the key insight. If H2NS is the hydrogen donor in bacteria, what's the oxidation product? Not oxygen downgrade, it's sulfur or sulfate. But in plants where water is the hydrogen donor, the oxidation product is oxygen. So the oxygen released in plants must come from water, not from carbon dioxide. This was proved later using radioisotopes Damp-14 and oxygen-18. So the oxygen in the atmosphere that we breathe, it comes from water in photosynthesis. So here's the complete accurate equation for photosynthesis in green plants. 6EOT plus 12H-tano with light produces 1 glucose NEET each. Plus 6 H-tano plus 6 OT. Notice something? We start with 12 water molecules and at the end we get 6 water molecules back plus 6 OT each. Why 12? Can you think about why? Because photosynthesis is not one simple reaction, it's a whole series of reactions we call a particular photosynthetic process. Different steps need different amounts of the raw materials. The oxygen released NEET that comes from those water molecules that were used. This has been proved using radioactive isotopes. So when we write this equation, we need to understand this is the overall summary of the entire process, which actually involves many individual reactions happening step by step. Okay, so where does photosynthesis happen? The most obvious answer is green leaves. But it's not just leaves. Any green part of the plant can do photosynthesis. Green stems, some flowers, buds, wherever there's green. Why green? Because that's where the chlorophyll is. That's where the chloroplasts are. Now inside the leaf, the mesophyll cells, the cells in the middle of the leaf, these cells have a huge number of chloroplasts. Lots and lots of them. These chloroplasts, they position themselves strategically. They align themselves along the walls of the mesophyll cells so they can get the maximum amount of incident light. When the light is bright and direct, they might position themselves perpendicular to the light rays. When it's low light, they might position themselves parallel to the walls to catch more light. It's automatic NEET. The plant optimizes light capture. Now let's go inside the chloroplast. Inside, there's a membranous system, membranes everywhere. These membranes stack to form structures called grana. You have stroma lamellae connecting them, and all of this is embedded in the matrix stroma, which is like the jelly inside the chloroplast. So this membrane system, what does it do? It traps light energy. This is where light reactions happen. This is where ATP and NADPH are made via the energy carriers. Then separately, in the stroma, the space inside, there are enzymes floating around. These enzymes do different reactions. They take the ATP and NADPH from the membrane reactions and they synthesize sugars. They make carbohydrates. So there's a clear division of labor here. Membranes, light reactions, stroma, sugar synthesis. Different jobs happening in different locations inside the same chloroplast. Clever, no? So we classify photosynthesis reactions into two types. Light reactions, photochemical reactions. These are directly driven by light. They happen in the membrane system. They produce ATP, NADPH and oxygen. Dark reactions, carbon reactions. These are not directly driven by light. They don't directly use light energy, but they depend on ATP and NADPH made by light reactions. They happen in the stroma. Now here's important. Don't think dark means these reactions happen in the dark, like in darkness. That would be wrong. The name dark just means they're not directly light dependent. By convention, we call them dark reactions. But really, they do depend on the products of light reactions, which only happen when there's light. So in complete darkness, dark reactions would stop eventually when the ATP and NADPH run out. Clear? Dark is just a conventional term, not describing when they actually happen. Ever notice how leaves aren't all the same shade of green? Even on the same plant, different shades? Why is that? We can answer this question using paper chromatography. Simple technique. You crush a leaf, make an extract, put it on filter paper, let it run through solvent. Different pigments move different distances. When you're done, what do you see? You see separate bands of different colors. And it turns out there are not one, not two, not three, but 4 different pigments in a leaf. First, chlorophyll amarynlis, that's the bright blue-green one. Second, chlorophyll B, yellow-green. Third, xanthophylls, jaune. Fourth, carotenoids, jaune to yellow-orange. All 4 together create the color we perceive. That's why leaves have these different shades of green. It's a mix of these 4 pigments in different proportions. Now what are pigments? Pigments are substances that can absorb light at specific wavelengths. Means what? Different pigments absorb different colors of light. So if I shine different colors on different pigments, each pigment absorbs some colors more than others. Look at chlorophyll A. When you measure its absorption spectrum throughout, which colors it absorbs? Both anthocytosol and archaeotroph have absorption in blue light and red light. In between, in the green region, it absorbs very little. That's why chlorophyll is green visible, because it's reflecting green light, not absorbing it. Now here's the interesting part. The action spectrum of photosynthesis throughout the wavelengths at which photosynthesis actually happens most activity in blue and red regions. So it matches the absorption spectrum of chlorophyll A. That means chlorophyll A is the chief pigment responsible for photosynthesis. But wait, if you compare very carefully, the absorption spectrum of chlorophyll A doesn't exactly match the action spectrum. There's overlap, but not complete one-to-one overlap. Why? Because there are these other pigments, accessory pigments, chlorophyll B, xanthophylls, carotenoids. These also absorb light, but in different wavelengths than chlorophyll A. They absorb light and then they transfer that energy to chlorophyll A. So chlorophyll A becomes the final acceptor, the one that does the photochemistry, but the energy comes from other pigments. This is very smart because now plants can use a wider range of wavelengths. Blue light, red light, yellow, orange, different pigments absorb different colors. And look what else, these accessory pigments protect chlorophyll A. Chlorophyll A is delicate, it can get damaged by too much light photooxidation, but accessory pigments absorb some of that extra energy, protecting chlorophyll A. So photosynthesis becomes more efficient and more protected. Good design, no? So when scientists measured the rate of photosynthesis across different wavelengths of light, what did they find? Action spectrum A, that's the graph showing photosynthetic rate. At different wavelengths, they found maximum photosynthesis in the blue region and in the red region. These match exactly with where chlorophyll A absorbs most. So this confirms chlorophyll A is the chief pigment. The pigment doing the main photochemical work is chlorophyll A. But if you look at the action spectrum more carefully, it's not zero at green wavelengths or yellow or orange. There's still some photosynthesis happening, lower than blue and red, but still happening. Why? Because those wavelengths are being absorbed by the accessory pigments. Xanthophylls absorb yellow, carotenoids absorb orange and yellow, chlorophyll B absorbs blue and red, but with different peaks than chlorophyll A. So when light of these wavelengths comes in, these pigments absorb it and pass the energy to chlorophyll A. It's like a relay. Different pigments catch different colors and they all pass the energy to chlorophyll A, which does the photochemistry. This is smart because it means plants can use a broader spectrum of light, not just pure red and blue, and the wavelengths in between. This makes photosynthesis more efficient in natural sunlight, which contains all wavelengths. Now let's understand how the photosynthesis machinery is organized. The pigments aren't just floating randomly, they're organized into specific units called light harvesting complexes or LHC. There are two main photosystems, photosystem I and photosystem II. Notice the naming, PSI and PS2. These are named in the sequence they were discovered by scientists, not in the sequence they function. That's important to remember. These photosystems are located in the thylakoid membrane. The LHC are made up of hundreds of pigment molecules, and these pigments are bound to proteins. Protein pigment complex is basically very organized. Each photosystem has almost all the pigments, chlorophyll A, chlorophyll B, xanthophylls, carotenoids, except one chlorophyll A molecule. That one special chlorophyll A forms what's called the reaction center. The rest of the pigments together form what's called the antennae or light harvesting system. Think of it like this. Hundreds of pigments are the antennae receiving radio signals. Light comes in at different wavelengths. These antennae pigments absorb it. The absorbed light energy bounces from pigment to pigment, eventually reaching the special chlorophyll A at the reaction center. At the reaction center, the actual photochemistry happens. Electrons get excited, reactions start. So the reaction center is where the work happens, but the antennae collect the light energy from all directions and deliver it there. This is very efficient. So each photosystem has a different reaction center chlorophyll. In photosystem I, the reaction center chlorophyll A has an absorption peak at 700 nanometers. That's in the far red region. We call this P700. P for pigment, 700 for the wavelength. In photosystem 2, the reaction center has an absorption peak at 680 nanometers. That's also red but not as far. We call this P680. So PSI has P700, PS2 has P680. Like one wavelengths, slightly different properties, both important for the reactions that follow. Okay, so let's trace the path of electrons. It starts in photosystem 2. The P680 reaction center absorbs red light at 680 nanometers. This energy excites the electrons at the reaction center. They jump to a higher energy level farther from the atomic nucleus. These excited high-energy electrons are unstable. They're picked up immediately by an electron acceptor. This acceptor passes them to an electron transport system containing cytochrome proteins. These proteins shuttle electrons from one to the next. As the electrons flow through this chain, they're going downhill in terms of energy piecemeal, or what scientists call redox potential. This downhill flow releases energy. This energy is important piecemeal. It's part of what makes ATP later. But right now, these electrons are being passed along, moving toward photosystem I. At the same time, in photosystem I, something similar is happening. The P700 reaction center absorbs red light at 700 nanometers. This excites the electrons at the PSI reaction center. These electrons are passed to an acceptor molecule that has an even higher redox potential. Then they flow downhill again, but this time to a molecule called NADPPS. These electrons reduce NADPPS plus. So now we have NADPH, an energy-rich molecule carrying electrons. This is one of the important products of the light reaction. NADPH will be used later in the dark reactions to help make sugars. Now let's put the whole picture together. Electrons start at PS2, get excited to a high energy level that's uphill. Then they flow downhill through the electron transport chain, passing through different cytochromes, losing energy, being caught by PSI. PSI electrons then get excited again, another uphill jump. Then they flow downhill again to NADP PAG. If you plot this on a graph showing energy levels with redox potential on the vertical axis, the path looks like the letter Z. That's why it's called the Z scheme. 2 uphill excitations, 2 downhill flows. Z-shape, this entire process. The Z scheme is how electrons are moved through the light reactions from PS2 all the way to forming NADPH. It's beautiful, really now. A coordinated series of reactions passing electrons through a chain, extracting energy at each step. So here's a puzzle. Electrons are being removed from photosystem 2, being excited, and then moved along. These PS2 electrons need to be replaced, otherwise PS2 cannot function. Where do the replacement electrons come from? The answer is water. Water is split. This is called photolysis of water or water splitting. 2 water molecules are split into 4 protons, 1 oxygen molecule and 4 electrons. These 4 electrons replace the 4 electrons that were removed from PS2. The protons go somewhere else. We'll talk about that. The oxygen gets released as O2 gas mass. That's the oxygen we breathe. Every oxygen molecule in the atmosphere came from water splitting in photosynthesis. Incredible, no? The water splitting complex is physically attached to photosystem 2. It's located on the inner side of the thylakoid membrane. Now here's a question. When water splits and produces protons and oxygen, where are these products released? Are they released in the lumen inside the thylakoid or on the outer side of the membrane? This is important because it affects where protons accumulate, which affects ATP synthesis later. But the key point is, the oxygen in the atmosphere comes from this water splitting, proved by radioisotope studies using oxygen-18. The oxygen atoms from CO10 don't appear in the O10 gas. Only the oxygen from water appears in the O10 we breathe. So remember, movie photosynthesis releases oxygen as a waste product. Oxygen is waste from the plant's perspective, but it's essential for us. The plant is purifying the air by splitting water and releasing oxygen. All living things need energy. We have food, plants have sunlight. But how is energy stored and used? Cells use special molecules to store energy in chemical bonds. The most important energy currency is ATP adenosine triphosphate. It has 3 phosphate groups bonded together. These bonds store a lot of energy. When the cell needs energy, it breaks one of these bonds. ATP becomes ADP adenosine diphosphate. Energy is released, available for cellular work. Now the cell needs to remake ATP from ADP. This process, adding a phosphate group back to ADP to make ATP kettles, is called phosphorylation. In cells, this happens in mitochondria during respiration and in chloroplasts during photosynthesis. In photosynthesis, the ATP synthesis process is called photophosphorylation. Photo means light, phosphorylation means adding phosphate. So it's the synthesis of ATP powered by light energy. This light energy is captured by the photosystems and it drives the whole process. In both photosynthesis and respiration, ATP synthesis is linked to something called a proton gradient. Photosynthesis, sense, same principle. This proton gradient is the key to ATP synthesis, and that's what we're going to explore next. During normal photosynthesis, there are 2 photosystems working together. PS2 first, then PSI. The electrons flow from PS2 through an electron transport chain to PSI, following the Z scheme we discussed. Both ATP and NADPH plus HPS are made by this process. This is called non-cyclic photophosphorylation. Non-cyclic because electrons don't cycle backwards. They flow forward from PS2 all the way to NADP-PS. Both energy molecules are produced ATP and NADPH. This happens primarily in the grana regions of the thylakoid membrane, where both photosystems are present. This is the main pathway, the one that keeps running during the day. It provides the ATP and NADPH that the Calvin cycle needs to make sugars. But wait, there's another option. Sometimes, if only photosystema is working prekinly PS meisten voses electron, then the electrons have a different path. They get excited at PSI, but they don't go to NADP price. Instead, they cycle back to PSI through the electron transport chain. The electrons go round and round, passing through cytochromes, getting pumped across the membrane, pumping protons. ATP is made because of the proton gradient, but NADPH is not made because electrons are not going to NADP price. This is cyclic photophosphorylation, cyclic because the electron cycle. This primarily happens in stromal lamellae and those are membrane structures different from grana. In grana, you have both PSI and PS2, so non-cyclic is happening. In stromal lamellae, PS2 is missing, so cyclic is what happens. Also, cyclic photophosphorylation occurs when you have only far-red light mesolectin beyond 680 nanometers. Why? Because PS2's P680 cannot be excited by wavelengths beyond 680. Only PSI's P700 can be. So if there's only far-red light, PS2 can't work and cyclic photophosphorylation is the option. So how exactly is ATP made? How do we go from light energy to ATP? The answer is something called the chemiosmotic hypothesis. It says that ATP synthesis is linked to a proton gradient, a difference in proton concentration across a membrane. In photosynthesis, the membrane is the thylakoid membrane. On one side is the stroma, on the other side is the lumen, the space inside the thylakoid. Now here's the key. Protons accumulate inside the lumen, meaning there are more protons inside than outside. This creates a gradient. The more inside, the steeper the gradient, the more potential energy is stored in that gradient. Now compare this to what happens in respiration. In mitochondria, protons also accumulate mass, but in the intermembrane space, not inside the matrix. Different location, same principle. Both use a proton gradient to make ATP. So where do these protons in the lumen come from? 3 sources. First source, water splitting. The photolysis of water happens on the inner side of the thylakoid membrane. When water splits, when suasnine, 4E my proso, but split. The protons are produced right there in the lumen. They accumulate there. Second source, electron transport. As electrons move through the photosystems, something interesting happens. At certain points, instead of just an electron carrier taking the electron, an H carrier takes it nitrogen carrier. This molecule removes the proton from the stroma from outside ion along with the electron. Then when it passes the electron to the next carrion on the inner side of the membrane, it releases the proton into the lumen. So protons are moved from stroma to lumen. Third source, NADP reduction. When NADP bias is being reduced to NADPH, many fact this happens on the stroma side. Protons are consumed from the stroma. Protons are taken from the stroma and incorporated into NADPH. So what happens overall? The stroma has fewer protons, they're being removed and sent into the lumen. The lumen has more and more protons from water splitting, from electron transport, from the H carriers. A gradient is established. High concentration inside, low concentration outside. You can actually measure this. The inside becomes more acidic, lower pH. The outside stays more basic, higher pH. This gradient, this concentration difference stores potential energy. It's like water behind a dam. You can release that energy to do work, and in this case, the work is making ATP. So we have this proton gradient, many protons inside the lumen, few protons in the stroma. Now the cell needs to use this gradient to make ATP. How? There's a special proton channel embedded in the thylakoid membrane. It's called ATP synthase. The CF counterpart, the CF0 is embedded in the membrane. It forms a channel. Protons are actively kept out of the stroma normally, right? But this channel allows them to flow back through facilitated diffusion. Protons flowing from high concentration to low concentration from inside the lumen to outside the stroma following the gradient. This flow is passive count. No energy needed, but the energy released as they flow is captured and used to make ATP. So ATP synthase has two parts. CF is in, the zerocus is in the membrane, it's the channel. CF1, the ventes, sticks out from the membrane on the stroma side. Think of it like a propeller. As protons flow through CF's roof, they cause CF1 to rotate. Actually, more accurately, as protons flow through, it causes conformational changes, Mal's shape changes, and CF1. These shape changes make CF1 work as an enzyme. And what does it catalyze? ATP synthesis. It takes ADP, adds a phosphate group, makes ATP. Multiple ATP molecules are made as the gradient flows through. Here's the key. As protons flow through the ATP synthase channel from the lumen to the stroma, the energy released causes CF meeting to change shape if the corpus the canopy is to unfrequency may change. When a protein changes shape, it can perform new functions. In this case, the shape change activates CF meeting as an enzyme that can add phosphate to ADP, making ATP. So the energy from the gradient is converted to mechanical energy shape change, which is then converted to chemical energy ATP. Multiple ATP molecules are synthesized. This is elegant. The light energy captured by the photosystems is used to pump protons. The protons want to flow back. That flow is harnessed to make ATP. Beautiful system. So let me summarize the whole chemiosmotic process. What do we need for this to work? 4 things. First, a membrane is a pseudopoid membrane which separates lumen from stroma. Second, a proton pump as something that moves protons across the membrane to create the gradient. That's the photosystems and electron transport chain. Third, a proton gradient, a difference in proton concentration. The gradient is created by water splitting, H carrier movement, and NADP reduction. Fourth, ATP synthase rounds, the protein that uses the gradient to make ATP. The cycle works like this. Light is captured. Energy pumps protons across the membrane. This creates a gradient. The gradient is a form of stored energy. ATP synthase taps this energy and lets protons flow back and harnesses that flow to make ATP. Brilliant design. Light energy proton gradient ATP energy. One more way to think about it. Imagine you're pumping water up a hill that takes energy. The energy comes from light in photosynthesis. Once the water is at the top, it has potential energy because it wants to roll back down. You put a water wheel at the bottom, that's ATP synthase. The water flowing down through the wheel provides energy to turn the wheel. The turning wheel grinds grain, that's making ATP. So in our system, light energy pumps protons uphill into the lumen. The protons want to flow back downhill. That's the gradient. ATP synthase is like the water wheel. Protons flowing through it drive it to make ATP. All along, the ATP that's made is used immediately. The dark reactions are waiting for it, waiting for ATP and NADPH to run the Calvin cycle and make sugars. So the light reactions make 3 products. First, ATP energy. Second, NADPH, a molecule with high energy electrons. Third, O2 oxygen gas. The oxygen, as we discussed, is released from water splitting. It leaves the chloroplast and goes into the atmosphere. From the plant's perspective, it's waste. But from our perspective, it's essential. It's the oxygen we breathe. The ATP and NADPH are not waste. They are immediately used. They don't stick around. They're taken to the stroma where the dark reactions, the Calvin cycle, are waiting for them. The dark reactions use both the energy from ATP and the electrons from NADPH to fix carbon dioxide and make sugars. So there's a direct pipeline. Light energy, ATP and NADPH, sugar synthesis. Continuous, coordinated process. Now we move to the second part of photosynthesis, the biosynthetic phase, also called dark reactions or carbon reactions. Now listen carefully. Dark doesn't mean it happens in the dark. It just means it's not directly driven by light. The biosynthetic phase doesn't directly use light energy, but it absolutely depends on the products of the light reactions ATP and NADPH. Without these, it cannot proceed. It needs COTO and HTMO as raw materials. It happens in the stroma of the chloroplast. And what does it make? Sugars, carbohydrates, glucose. The whole point of photosynthesis is to take carbon dioxide and water and make sugars. That happens here in the biosynthetic phase using the energy from ATP and electrons from NADPH. So how can we prove that dark reactions depend on light reaction products? Simple experiment. You have a plant doing photosynthesis. Light is on, synthesis is happening. You turn off the light. What happens to sugar synthesis? It continues, but only for a little while. Then it stops. Why? Because there's still ATP and NADPH in the stroma from the light reactions that just happened. The dark reactions keep going, using up these energy molecules. But once these are depleted, no more ATP, no more NADPH, synthesis stops. Now turn the light back on. The light reactions start again, making ATP and NADPH. The dark reactions start again immediately. This proves it and the dark reactions depend on the light reaction products, not directly on light, but on ATP and NADPH. So calling it dark is misleading. The reactions don't really happen in the dark. They would stop without light because light is needed to regenerate ATP and NADPH. After World War II, scientists wanted to use radioisotopes for peaceful purposes. One scientist who did brilliant work was Melvin Calvin. He took the idea of radioactive carbon-14, a radioactive isotope, and used it to trace what happens in photosynthesis. He worked with algae, chlorella. He exposed the algae to CO2 containing carbon-14. Then at different time points, he stopped the photosynthesis, extracted the molecules, and identified them. The carbon-14 would show him which molecules contain the carbon dioxide. By doing this systematically, he traced the path of carbon through the photosynthetic reactions. What he found? The first stable product when CO2 is fixed into an organic molecule is a 3-carbon compound NAX, a 3-carbon acid. This was breakthrough work. Calvin didn't just find one product, he worked out the entire pathway. He identified all the intermediate compounds, the sequence of reactions. Because of his contributions, the entire pathway was named after him, the Calvin cycle. The first product in the cycle is 3-phosphoglyceric acid, abbreviated PGA. PGA has 3 carbon atoms, so it's a 3-carbon compound. Remember that we mimics, it's important. The pathway itself is also called the CTAR pathway because the first product has 3 carbons. This was the beginning of understanding how photosynthesis actually works at the molecular level. Before this, people knew the overall equation, but not the actual reactions. Calvin revealed the reactions, step by step. After Calvin's work, scientists asked, do all plants have the same photosynthetic pathway? Does every plant have PGA as the first product? They tested many plants from different environments, different countries, different climates. Surprisingly, they found that not all plants make PGA first. Some plants, when exposed to CO2 and allowed to photosynthesize briefly, produced a different first product. Instead of a 3-carbon compound, they produced a 4-carbon compound, acidic acid, abbreviated OAA. This was fascinating. 2 different pathways, 2 different first products. So scientists classified plants into 2 groups based on this. Side plants, the first product has 3 carbons PGA. C-palm plants, the first product has 4 carbons OAA. These weren't just chemical differences. The 2 groups had other differences too, in leaf structure, in enzyme distribution, in temperature preference, in where they grow naturally. The C-palm plants were found everywhere. The C-palm plants were mostly found in tropical and subtropical regions, in hot, dry areas. These pathways reflected different evolutionary solutions to photosynthesis, different strategies that plants had developed to succeed in their particular environment. So here's a puzzle Calvin faced. The first product is PGA3 carbons. One of those carbons comes from CO10, carbon. So the acceptor molecule, the molecule that receives the CO3, must have had 2 carbons originally. This seems logical, no? Scientists thought so too. They searched and searched for a 2-carbon molecule. Years and years of work. No luck, it didn't exist. This is science sometimes. Sonderkical hypothesis is wrong. Then the answer came and it was unexpected. The acceptor molecule is not a 2-carbon compound. It's a 5-carbon compound. A 5-carbon sugar ribulose bisphosphate RuBP. How does this work? When 5-carbon RuBP reacts with 1-carbon COTC, it forms an unstable 6-carbon intermediate that immediately splits into 2 molecules of 3-carbon PGA. So the math works out. 5-carbon, 1-carbon, 2 carbons. Carbon. 5? But who would have thought the acceptor is 5 carbons? Scientists took a long time and did many experiments to figure this out. And that's okay. That's how science works. You propose, you test, you get unexpected results, you revise your understanding. Eventually, you reach the truth. So the Calvin cycle, the dark reaction pathway, has been broken down into 3 stages to make it easier to understand. First stage is carboxylation, that's the fixation of CO10. CO10 gets incorporated into a stable organic molecule. Second stage is reduction. That's where the fixed carbon gets converted toward glucose. It's called reduction because electrons are added. Third stage is regeneration, that's where the CO10 acceptor molecule RuBP is regenerated so the cycle can continue. The cycle is cyclic. It goes round and round. CO10 is fixed, something is made, and then the starting molecule is regenerated ready for the next cycle. This 3-stage framework helps organize the many reactions that happen. Here's an important point. Please listen carefully. The Calvin cycle occurs in all photosynthetic plants. All of them. Whether a plant is C. tara or C. tara, it has the Calvin cycle. Whether it's a corn plant, a wheat plant, a tomato plant, the Calvin cycle is there. The difference between C. tara and C. tara plants is not whether they have the Calvin cycle. The difference is in what happens before the cycle starts. And the difference is in where the cycle happens, in which cells. But the Calvin cycle pathway itself, same in all plants. Universal. This is crucial to understand. The C. tara and C. tara classification is not about the Calvin cycle. It's about the initial CO10 fixation. The carboxylation stage, this is the most crucial step. This is where atmospheric carbon dioxide gets captured and incorporated into an organic molecule. Here's what happens. RuBP, the 5-carbon acceptor molecule, meets CO10. They react. For a brief moment, they form a 6-carbon intermediate compound. This is unstable. It immediately splits into 2 molecules of 3-PGA maize 3-carbon compounds. So we've taken 1 CO10 from the atmosphere and incorporated it into organic molecules. 1 carbon from the air is now part of the plant. This is the foundation of photosynthesis, capturing atmospheric carbon and making it into plant material. And eventually, all food comes from this step. The enzyme that catalyzes this reaction is RuBP carboxylase. But wait, this enzyme can do 2 things. It can add CO2 succinylation or it can add OT oxygenation. So the complete accurate name is RuBP carboxylase oxygenase succinylane, RuBCO. We'll talk later about what happens when it adds OT instead of COT. But for now, understand. RuBCO catalyzes the carboxylation reaction. RuBP plus COT produces 2PG. And here's something fascinating. RuBCO is the most abundant enzyme in the world. It's the most abundant protein. In fact, about 50% of all protein in the green leaf is RuBCO. Why? Because it's needed in huge quantities to fix all the CO2 needed for photosynthesis. This one enzyme is the foundation of life on Earth. It's the enzyme that captures atmospheric carbon. The second stage is reduction. Remember, reduction means adding electrons. Here, the PGA molecules that were made in carboxylation are now reduced to make glucose. This requires energy, lots of it. For every CO2 molecule fixed, you need 2 ATP molecules. Why ATP? Because reactions need energy to proceed. Phosphorylation reactions especially need that energy from ATP. You also need 2 NADPH molecules per CO2. NADPH carries electrons. Reduction means adding electrons. So NADPH provides those electrons. Both ATP and NADPH come from the light reactions. This is why the light reactions are so important. They provide the energy and the electrons needed for the dark reactions to proceed. Without ATP and NADPH from light, no reduction, no glucose. So let's do the math. To make one glucose molecule which has 6 carbons, you need to fix 6 COT molecules. The Calvin cycle must turn 6 times. If each COT needs 3 ATP weight, we'll talk about the third ATP in regeneration market. But for reduction specifically, 2 ATP per COT. That's 2 ATP, 6, 12 ATP for reduction. And 2 NADPH per COT. That's 2E612 NADPH. But wait, the regeneration stage also needs ATP, 1 ATP per COT, that's 6 more ATP. So total 18 ATP. Got it? 18 ATP and 12 NADPH to make 1 glucose. These numbers are important. You should know them. They show how energy intensive photosynthesis is. But it's worth a click. We get glucose, we get energy, we get food. Third stage, regeneration. This stage is all about regenerating the starting molecule RuBP, so the cycle can go again. The molecules produced in the previous stages are rearranged through complex reactions. In this, there are many intermediate steps. These reactions require energy, specifically ATP. 1 ATP per CO10 is used to phosphorylate the intermediates to form RuBP. Why is regeneration important? Simple move. If you don't regenerate RuBP, what happens next? CO10 comes in, but there's no acceptor for it. The cycle stops. So regeneration isn't optional, it's essential. It's the price you pay to keep the cycle running. You need to invest ATP to keep the cycle turning. Now when we add up all 3 stages for 1 COT, carboxylation doesn't use ATP directly. Reduction uses 2 ATP. Regeneration uses 1 ATP. Total 3 ATP per COT and 2 NADPH in reduction. For 1 glucose nil, 6 COT notch, that's 18 ATP and 12 NADPH. These numbers, remember them. You see, during non-cyclic photophosphorylation, equal amounts of ATP and NADPH are made. But the Calvin cycle needs more ATP than NADPH. ATP and NADPH ratio needed is 3.2, but non-cyclic gives 1:1. So what happens? Cyclic photophosphorylation produces extra ATP without making NADPH. That's why cyclic photophosphorylation is needed now to balance the energy supply with the energy demand of the Calvin cycle. Beautiful system. So let's summarize the Calvin cycle. What goes in? 6 molecules of CO2 from the atmosphere. That's the raw material we want to capture. 18 ATP molecules energy from the light reactions. 12 NADPH molecules, electrons, from the light reactions. These energy molecules come from the light reactions that happened in the thylakoids. What comes out? 1 glucose molecule, that's the goal. The ATP has been used up. It becomes ADP and gets sent back to the light reactions to be regenerated. The NADPH has been used. This put which becomes in NADPH and goes back to the light reactions to be reduced again. So the cycle takes things from the light reactions, uses them to make glucose, and the energy molecules go back to the light reactions to be recharged. Continuous cycle, continuous energy, beautiful system, everything is recycled. Our freight yarns are adapted to dry, tropical regions. They get their name because their first CO10 fixation product has 4 carbons, oxaloacetic acid. But here's important, they still use the Calvin cycle. They're not doing something completely different. Instead, the C-core pathway happens first. Then the product goes to the Calvin cycle. So it's not C-core. Instead of C-core, it's C-core and then C-core. The C-core part is a preliminary step that sets up the Calvin cycle to work better in these hot, dry environments. Why would plants evolve this? Because it gives them advantages in those harsh conditions. So what makes sea proud plants special? 5 main things. First, they have special leaf anatomy B. We'll see this in detail. Second, they can tolerate much higher temperatures than sea proud plants. Third, they respond strongly to high light intensities B. They can use intense light efficiently. Fourth, and this is important, they lack photorespiration. We'll talk about photorespiration later, but it's wasteful. Sea proud plants don't have this problem. Fifth, they have greater biomass productivity. They grow more, produce more. This explains why corn, sorghum, sugarcane, maize, all sea proud plants are so productive in hot climates. This explains why they're better crops in tropical and subtropical regions. It's not just chance, it's biology. Evolution has adapted these plants perfectly to these environments. In sea plants, there's a special leaf structure. Around each vascular bundle, the veins of the leaf, there are large cells called bundle sheath cells. These cells form layers, multiple layers sometimes, like a wreath around the bundle. That's why the overall structure is called Kranz anatomy. Kranz is German for wreath. The arrangement looks like a wreath. This is not present in sea plant plants. When you look at a sea plant leaf under the microscope, you see normal mesophyll tissue. But when you look at a C-clone plant leaf, you see this distinct ring of large cells around the bundles. This is a key identifying feature. These bundle sheath cells have special properties. They contain a huge number of chloroplasts, much more than normal mesophyll cells. Their cell walls are thick, and importantly, these walls don't allow gases to pass through. No gas exchange, no intercellular spaces. So gases like CO2 cannot diffuse easily between the bundle sheath cells and the mesophyll cells around them. This might seem like a disadvantage, but it's actually the key to why C-clone plants work so well. By compartmentalizing separating the mesophyll from the bundle sheath different reactions can happen in different places. Efficient system. You can observe this yourself. Take a leaf from a corn plant or sorghum plant. Both C-clone plants cut a thin vertical section through the leaf, put it on a microscope slide, look at it under the microscope. You'll see the veins, the vascular bundles. Around each vein, you'll see large cells forming layers. That's Kranz anatomy. Compare this to a tomato leaf or a bean leaf, C-clone plants. The structure is completely different. The mesophyll is just scattered around the vein, no special organization. So if you want to identify whether a plant is C-clone or C-clone, cut a leaf section and look for bundle sheath cells. If they're there organized in layers around the vein, it's a C-clone plant. Simple identification method. The C-clone pathway has a special name, the Hatch and Slack pathway, named after the scientists who discovered it. It's a cyclic process. The first step happens in the mesophyll cells hasher pistules, the cells in the middle of the leaf. Here, CO10 is fixed. But the acceptor molecule is different from the Calvin cycle. Instead of ruby P, it's a 3-carbon molecule called phosphanol pyruvate, abbreviated PEP. This PEP is already present in mesophyll cells. Now CO10 combines with PEP. The enzyme doing this is PEP carboxylase neurone. You might hear it called pepcase. This enzyme is present in mesophyll cells. Here's the key distinction. Rubisio is not present in mesophyll cells. So carboxylation in C-clone plants doesn't use rubisio, it uses pepcase. From PEP plus COTD, you get OAA-hexoxyoacetic acid, a 4-carbon compound. This is why it's called the C-clone pathway. The first stable product is 4 carbons. This is completely different from C-clone plants, where rubisio is in mesophyll cells and makes PGA directly. So OAA is made in the mesophyll. But OAA is not the end product. It's converted into other 4-carbon compounds. Malic acid is common, aspartic acid is another. These are still 4-carbon molecules. All of this is happening in the mesophyll cells. Then these 4-carbon compounds are transported. They move from the mesophyll cells into the bundle sheath cells. This transport happens through plasmodesmata, the connections between plant cells. Remember, we said bundle sheath cells have thick walls and no intercellular spaces. They must communicate somehow. They do through plasmodesmata. The 4-carbon compounds travel through these connections from mesophyll to bundle sheath. Once these 4-carbon compounds reach the bundle sheath cells, something important happens. They're broken down, decarboxylated. A CO10 molecule is released. When you remove 1 carbon as CO10 from a 4-carbon molecule, what's left? A 3-carbon molecule. So now, there's CO10 being released right there in the bundle sheath cells, where the Calvin cycle machinery is. And the 3-carbon product goes back to the mesophyll to regenerate the cycle. But the important thing is the CO10 concentration. The bundle sheath cells now have a very high concentration of CO10 right where rubisio is located. This is the advantage of the C-car pathway high CO10 concentration at the enzyme site. The 3-carbon molecule, what's left after CO10 is released, goes back to the mesophyll. It doesn't stay in the bundle sheath, it travels back through plasmodesmata to the mesophyll cells. In the mesophyll, this 3-carbon molecule is converted back into pepiphosphanol pyruvate. This conversion requires multiple steps and requires ATP. Energy is invested to regenerate pepi. Now pepi is available again to accept another CO10 molecule. The cycle is complete. Ready to do it again. So the C-carbon mishram, its circular cycle is cyclic. It's circular. CO10 is fixed to pepi, producing 4-carbon compounds, transported to bundle sheath. CO10 is released, 3-carbon goes back, pepi is regenerated, round and round. Now the CO10 is released in the bundle sheath. This enters the Calvin cycle. The Calvin cycle happens only in bundle sheath cells in C-core plants. This is different from C-core plants where Calvin cycle is in mesophyll. So in C-core plants, the bundle sheath cells are packed with rubisio, sniffle, lots of rubisio, but no pepcase. The mesophyll cells have pepcase but no rubisio. Complete compartmentalization, division of labor. Mesophyll does the initial C-core fixation and transport. Bundle sheath does the final fixation and sugar synthesis. The C-corp concentration in bundle sheath is high from the C-corp compounds being broken down. High C-corp NAN at the site of Rubisio means efficient photosynthesis, and that's the whole strategy of C-corp plants. Let me compare C-par and C plants. First, difference, the acceptor molecule. In C-par plants, CO2N is accepted by RuBP, a 5-carbon molecule. In C-par plants, CO2N is accepted by PEP means a 3-carbon molecule. Because of these different acceptors, the first products are different. In C-par plants, CO2N plus RuBP makes PGA mix 3 carbons. In C-par plants, CO2N plus PEP makes OAA mix 4 carbons. This is where the names come from. C-par pathway because first product has 3 carbons. C-core pathway because first product has 4 carbons. Second difference, enzyme distribution. In C5 plants, Rubiscio is in the mesophyll cells. That's where carboxylation happens. C5 plants don't have pepcase. In C5 plants, pepcase is in mesophyll nucleus. That's where the initial CO10 fixation happens. Rubiscio is not in mesophyll, it's in the bundle sheath cells. So the enzymes are compartmentalized. Different cells have different enzymes. This creates the division of labor unique to C5 plants. Third difference, and this is crucial, Calvin cycle location. In C. clara plants, the Calvin cycle happens in the mesophyll cells all throughout. In C. clara plants, the Calvin cycle does not happen in mesophyll cells. It only happens in bundle sheath cells. This is a fundamental difference in how the plant is organized. Now remember, both C. clara and C. clara plants have the Calvin cycle. It's not like C. clara plants don't use Calvin cycle. They do, but the location is different. C. clara uses it in mesophyll, C. clara uses it in bundle sheath. This compartmentalization in C. clara plants is what gives them their advantages. Let's talk about something that happens in C. col plants but not C. col plants. This photorespiration. This is important because it explains a major difference between the two plant types. Photorespiration happens at the very first step of the Calvin cycle, the carboxylation step. The enzyme Rubiscio has just done its job. It fixed CO2N, made 2 PGA. Normal, but sometimes something different happens. Sometimes instead of C-col1, oxygen is added, and that leads to a different reaction, a different product, a wasteful product. Remember the enzyme's full name, RuBP carboxylase oxygenase? The name tells you something. Carboxylase, it can add carboxyl groups, meaning CO to it. Oxygenase, it can add oxygen, O to it. The active site of this enzyme is flexible. It can bind either CO to it or O to it. Both molecules are roughly similar in size and shape. So the enzyme's active site can accommodate either one. That's why the enzyme has this dual activity. So here's the situation. RuBP is there, CO2N and O2N in the air compete for the Rubiscio active site. It's competitive binding. Which one gets there first? It depends on their relative concentrations. Now Rubiscio does have greater affinity for CO2N. Wick prefers CO2N when the concentrations are roughly equal. But in reality, O2N is much more abundant in the atmosphere than CO2N. CO2N is only 0.03 to 0.04%. O2N is 21%. So even though Rubiscio prefers CO2N, O2N is so much more abundant that quite a bit of O2N binds instead. In CO2N plants, this is a problem. In C-pary plants, O2N does succeed sometimes. It binds to Rubiscio. When this happens, CO10 fixation decreases because O2N took the binding site that CO10 could have used. In the normal carboxylation reaction, RuBP plus CO2N gives 2 molecules of PGA. But when oxygenation happens, RuBP plus O2N gives 1 3-carbon phosphoglycerate and 1 2-carbon phosphoglycolate. The products are different. You've lost efficiency, you've consumed RuBP but not fixed carbon mash, you've fixed oxygen instead. This entire process of O2F binding instead of CO2F leading to different products, this is photorespiration. It's called photorespiration because it only happens in the light when photosynthesis is happening. And it looks like respiration in some ways. It consumes organic molecules. But it's not respiration, friends. It's a branch pathway from photosynthesis. The consequence, your overall photosynthetic rate goes down. You wanted to make sugar, but instead, some of your RuBP is being wasted on fixing oxygen. This becomes especially a problem in hot, dry conditions where O2F CO2F ratio is high. So what happens in the photorespiratory pathway? First, there's no sugar synthesis. You're not making glucose. Second, there's no ATP synthesis. ATP is not being made. Third, ATP is being consumed, used up. Fourth, CO2N is released. So you're losing CO2N. This is the opposite of photosynthesis. Photosynthesis makes ATP, makes sugars, fixes CO2N. Photorespiration consumes ATP, makes nothing useful, releases CO2N. It's wasteful, pure loss. A plant would much rather avoid this if it could. In the photorespiratory pathway, there's neither ATP synthesis nor NADPH synthesis. Only consumption, no production of these energy molecules. So it's purely wasteful from an energy standpoint. Now here's an interesting question. Why does this pathway exist at all? Why would evolution keep this? Scientists don't know yet. The biological function of photorespiration is still a mystery. Some hypotheses exist. Maybe it's a leftover from evolution. Maybe it serves some regulatory function. Maybe there's some advantage we haven't discovered yet. But for now, it remains unexplained. All we know is that it's wasteful and plants would be better off without it. Here's the genius of sea ocean plants. They have a mechanism to prevent photorespiration. The mechanism is simple. Increase the sea ocean concentration at the site where rubis-yo is working. Here's how. The 4 carbon acids from the mesophyll are transported to the bundle sheath cells. In the bundle sheath, these acids are broken down decarboxylated. CO chain is released. This CO chain is released right there in the bundle sheath, right where the rubis-yo is located. So there's a huge concentration of CO chain right at the enzyme site. At high CO chain concentrations, rubis-yo strongly prefers carboxylation over oxygenation. The high concentration of CO chain outcompetes the OTA in the atmosphere. OTA cannot bind because all the enzyme sites are occupied by CO chain. So in bundle sheath cells, rubis-yo functions primarily as a carboxylase is the COT adding function. The oxygenase activity is minimized. Even though OT is present, even though the enzyme can add OT, it's so busy adding COT that very little oxygenation happens. Photorespiration essentially doesn't occur in sequoia plants. This is major because all the energy of photosynthesis goes into making sugars, not into this wasteful photorespiratory pathway. Sequoia plants are efficient. Now you understand why sequoia plants are so much more productive. They don't waste energy on photorespiration. Every ATP, every NADPH produced by the light reactions goes into productive work, making sugars, making biomass. Sequoia plants, on the other hand, are losing energy constantly to photorespiration. Some of their ATP is consumed without making anything useful. Some of their RuBP is wasted. So sequoia plants have better productivity, higher yields, better photosynthetic efficiency. From an agricultural perspective, this is huge. Also, sea plants tolerate higher temperatures much better than sea plants. Sea plants have a lower temperature optimal. They like it cooler. Sea plants can handle heat. This makes them perfectly adapted to tropical and subtropical climates, hot, dry regions. That's where sea plants thrive. This is why corn, sugarcane, sorghum, all sea crops are grown in hot regions. It's not just a coincidence, it's biology. These plants evolved in hot climates and they have mechanisms adapted to hot conditions. No photorespiration, higher temperature tolerance. So if you're farming in a hot region, you choose sea plants. If you're in a temperate climate, sea plant crops might be fine. Different plants, different strategies, different environments. Nature has adapted plants to their conditions. So we're talking about what limits photosynthesis. There are factors within the plant itself that affect how much photosynthesis happens. Plant factors. First, the leaves themselves. How many leaves does the plant have? How big are they? How old are they? The age matters. Young leaves might not photosynthesize as efficiently as mature leaves. Leaf orientation also matters. How the leaves are positioned relative to the sun. Second, the mesophyll cells, their characteristics affect photosynthesis. Third, the chloroplasts. How many does each cell have? What type are they? Fourth, internal CO10, the concentration of CO10 inside the leaf. Fifth, chlorophyll. How much chlorophyll is present in the leaves? All these factors are plant factors, internal factors. Now these plant factors, how many leaves, how big they are, how many chloroplasts, how much chlorophyll, these all depend on genetics and growth. Genetics determines what's possible. A corn plant is genetically programmed to be a certain height, to have a certain leaf size, to produce a certain amount of chlorophyll. Growth and development determines where the plant is now. Is it young? Is it mature? Is it stressed? A stressed plant may not produce as much chlorophyll. A young plant may not have as many leaves as it will have eventually. So the internal plant factors depend on both genetic predisposition, what the plant is capable of, and the growth state. What stage is the plant in right now? These factors are relatively fixed in the short term. You can't suddenly increase the number of leaves on a plant or the number of chloroplasts. These take time, growth. Now let's talk about external factors, environmental factors outside the plant. Four main ones. Sunlight, how much light is available. Temperature, how hot or cold is it. CO2 in concentration, how much carbon dioxide is in the air. Water, is water available to the plant. All of these affect photosynthesis and all of them can change. Throughout the day, sunlight changes. Season to season, temperature changes. After a rain, water availability changes. CO2 in concentration usually doesn't change much day to day, but it can vary with location and air movement. So these external factors are variable, changeable, affecting photosynthesis in real time. Here's the thing about external factors. They all act simultaneously. The plant isn't just responding to light, it's responding to light and temperature and CO2 and water at the same time. These factors interact. Good light but low temperature, photosynthesis might be limited by temperature. Good temperature and light but low CO2, limited by CO2. Multiple factors are in play, but at any given moment, usually one factor is the most limiting. The factor that's at suboptimal levels, that's the factor that determines the overall rate. If all factors are optimal except CO2, which is low, then CO2 is limiting. The rate is determined by the most limiting factor. This is important to understand because it helps farmers optimize crop growth. A scientist named Blackman in 1905 figured out a principle that applies to photosynthesis and to any process affected by multiple factors. It's called the law of limiting factors. It says, when a chemical process is affected by more than one factor, the rate of the process is determined by the factor which is nearest to its minimal value. Means what? The factor that's most deficient, the factor that's most lacking. That factor determines the rate. All the other factors could be abundant, but if one is lacking, that's the bottleneck. That determines how fast the process goes. Here's an example. You have a nice green leaf. Perfect. You put it in optimal light, lots of light, exactly what's needed. You have optimal CO2 concentration. Everything is perfect, but the temperature is very low. What happens? Does the plant photosynthesize at a normal rate? No, it doesn't. Why? Because temperature is too low. Temperature is the limiting factor. Even though everything else is optimal, temperature is holding things back. It's the bottleneck. Now to warm it up, you increase the temperature to optimal levels. Suddenly the plant starts photosynthesizing at a normal rate again. Why? Because you remove the limitation. The limiting factor is no longer limiting. This is Blackman's principle in action. Powerful concept for understanding photosynthesis and growing plants. When we talk about light as a limiting factor for photosynthesis, we need to be specific. There are actually three different aspects of light we should distinguish. First, light quality, that's the wavelength, the color. Blue light, red light, green light, infrared, different wavelengths. Second, light intensity, that's the brightness, how much light energy is coming in. Strong light versus weak light. Third, light duration, that's how long the light is present. 8 hours of light versus 12 hours versus 24 hours. These three are different. They're not the same thing. When we talk about light limitations, we need to know which aspect we're talking about. Is it the color that's wrong? The brightness? The length of exposure? Different answers for different situations. So when you measure photosynthetic rate at different light intensities, what do you find? At low light intensities, there's a nice linear relationship. More light means more photosynthesis. If you double the light, you roughly double the photosynthetic rate. Linear. Proportional. Straight line on the graph. This makes sense. Light is the limiting factor at low intensities, so more light directly increases the rate. But at higher light intensities, something different happens. You keep increasing the light, but the photosynthetic rate doesn't increase anymore. It plateaus, it levels off. Why? Because light is no longer the limiting factor. Something else is limiting now. Maybe it's temperature too hot or too cold. Maybe it's CO2 concentration, not enough CO2 in the air. Maybe it's something else. But whatever it is, increasing light won't help anymore. The rate is determined by the other limiting factor now. This is Blackman's principle in action. At high light, light is not the limit, so giving more light doesn't help. On the graph, this shows as a horizontal line no change in rate, even as light continues to increase. Here's an important fact. light saturation occurs at about 10% of full sunlight. Think about that, 10%. So plants are saturated with light when they receive just 10% of the full sun's intensity. What does this mean? For most plants in outdoor conditions, light is not a limiting factor. They're already receiving more light than they need. Except for plants that live in deep shade under a dense forest canopy, for example or plants in really dark caves, most plants are not light limited. They receive plenty of light. This changes our thinking. It means that in nature, the limiting factors are usually not light. It's probably temperature or CO2 in water. Not light. Light is abundant. But here's another important point. Too much light can be bad. If you increase light intensity beyond optimal levels, photosynthesis doesn't keep increasing forever. At some point, excessive light causes damage. The chlorophyll gets broken down, that is photooxidation. The photosynthetic machinery gets damaged, and the photosynthetic rate actually goes down. So while light at low levels is usually not limiting, too much light is harmful. There's a sweet spot, optimal light. Neither too little nor too much. This is important for agriculture. You want to provide good light, but not so much that it damages the plants. Carbon dioxide, this is the major limiting factor for photosynthesis. Why? Because CO2 is scarce. In the atmosphere, CO2 is only 0.03 to 0.04%. That's very low. 0.03 to 0.04%. Compare that to oxygen, 1%. Water vapor jama or O can vary, but often several percent. But CO2, 0.03 to 0.04%. Scarce. So plants are often limited by CO2. There's not enough of it. Scientists found that if you increase CO2 concentration up to 0.05%, that's higher than natural levels, photosynthesis increases. The fixation rate goes up. If you go beyond 0.05%, it becomes harmful. The plant gets stressed. So the optimal range is maybe 0.04 to 0.05%, and anything higher starts to be damaging. The point is, CO2 is what limits photosynthesis in most situations. So C-pyre C plants, do they respond the same to CO2's concentrations? Let's see. First, at low light conditions. Neither plant type responds to high CO2. Why? Because light is limiting. No point having extra CO2 if there's no light to drive photosynthesis. So both types plateau at low CO2 when light is low, but at high light intensities. Now both types respond. Both show increased photosynthesis with increased CO2. Both respond to more carbon dioxide. Where they differ is in how much CO2 they need before they saturate. Seaport plants saturate at about 360 microliters per liter. That's lower than seaport plants. Seaport plants don't saturate until beyond 450 microliters per liter. Current atmospheric CO2 is about 0.04%. That's around 400 microliters per liter. So current CO2 levels are pretty close to seaport saturation needs. They're doing fine. But for seaport plants, current levels are definitely below their saturation point. Seaport plants are limited by current CO2 levels. If you gave them more CO2, they'd photosynthesize faster. This is why CO2 enrichment in greenhouses is beneficial for seaport crops like tomatoes and peppers. You increase CO2, the plants photosynthesize faster, you get higher yields. It's an agricultural application of understanding photosynthesis limiting factors. So here's how this knowledge is actually used in agriculture. Scientists know that seaport plants respond to higher CO2 by photosynthesizing faster. So greenhouse growers take advantage of this. They pump extra CO2 into their greenhouses. This enriches the atmosphere inside the greenhouse with CO2. The CO2 concentration becomes higher than outside. Now the plants inside, if they're seaport plants, they photosynthesize faster. They produce more sugars, more growth, more fruit, higher yields, better productivity, more profit. Specific examples, tomatoes and bell peppers. Both seed-type plants grown in greenhouses in many countries. The growers maintain elevated CO2 levels inside the greenhouse. Compared to outdoor plants where CO2 is ambient, these greenhouse plants have more CO2 available. They photosynthesize faster, they grow more, they produce more fruit, higher yields. This is real-world application of what we learned in this chapter. Understanding that CO2 limits seed-type plant photosynthesis and that increasing CO2 increases photosynthesis led to a practical technique that increases agricultural productivity. Theory becomes practice, science becomes profit. Temperature is another limiting factor, and here's the biochemical basis. The dark reactions are enzymatic. They depend on enzymes like rubisio to proceed. Enzymes are temperature dependent. At low temperature, enzymes work slowly. As temperature increases, Enzymes work faster. There's an optimum temperature where the enzyme works best. Go higher than that. The enzyme denatures, it loses its shape, and activity drops. So dark reactions have a temperature optimum. Below it, they're slow. Above it, they're also slow. The light reactions are also temperature sensitive, but not as much. Light reactions involve photochemistry. Light is driving them, so temperature affects them less than it affects purely enzymatic reactions. And here's where sea plants differ. Sea plants respond to higher temperatures. Higher temperature, higher photosynthesis rate. Sea plants have a higher temperature optimum. They like it hot. Sea plants have a lower temperature optimum. They prefer cooler conditions. Higher temperature actually slows down their photosynthesis. This explains why sea plants are found in hot regions cornered where if they adapt to heat, sea plants kichlu. Sea-type plants are found in temperate regions where they adapted to cooler conditions. Evolution has fine-tuned each plant type to its climate. Sea prior to heat and drought, sea prior to moderate temperatures. Temperature optimum for photosynthesis. It's not the same for all plants. It depends on where the plant comes from. Tropical plants that evolved in hot climates, they have high temperature optimum. Their photosynthesis works best at higher temperatures. Temperate plants that evolved in cooler climates, they have lower temperature optimum. Their photosynthesis works best at lower temperatures. Why? Evolution, natural selection. Over many generations, plants that were better adapted to their local temperatures succeeded and reproduced. Plants that weren't adapted well didn't survive as well. So the photosynthetic machinery gradually got fine-tuned to the local climate. Tropical plants got tuned for heat. Temperate plants got tuned for cooler conditions. This is adaptation, evolution shaping plants to their environment. And this connects to what we learned about sea part and sea plants. Many tropical plants are sea part plants with high temperature optimum. Many temperate plants are sea part plants with lower temperature optimum. It's not a coincidence, it's integration. A plant doesn't just have one adaptation to its climate, it has multiple adaptations working together. Sea part pathway, high temperature optimum, photorespiration avoidance C, these all work together to make the plant suited to hot, dry tropical conditions. Sea part pathway, lower temperature optimum, different leaf anatomy, these work together to suit temperate conditions. Evolution builds complete packages suited to particular environments, and different plants in different parts of the world have these different packages. Water is a reactant. In the light reaction, water gets split, producing electrons and oxygen. So what is needed? But here's the thing, it's usually not the limiting factor for photosynthesis, even though it's a required reactant. Why? Because the plant has plenty of water unless there's a drought. The direct effect of water as a chemical reactant is rarely the limit. Instead, water acts as a limiting factor through indirect effects. When water becomes scarce, drought conditions, the plant responds. The plant closes its stomata to conserve water. The stomata are the pores in leaves that allow gases to exchange. Closing them saves water because water doesn't evaporate through the pores. But closing stomata also prevents CO2 from entering the leaf. So now the plant is limited by low CO2 inside the leaf, not by water directly. The water stress caused the CO2 limitation indirectly. And there's more. Water stress also causes leaves to wilt. When water is scarce, cells lose turgor output. Water pressure in the cells. Leaves droop, lose their rigid structure. Drooping leaves have less surface area exposed to light. Less surface area means less photosynthesis. Also, the overall metabolic activity of the plant decreases under water stress. The whole plant slows down. Photosynthesis slows down as part of this general slowdown. So water scarcity affects photosynthesis in multiple ways. Stomatal closure, reducing CO2, wilting, reducing surface area, general metabolic slowdown. All indirect effects. None of these are directly about water as a chemical reactant. They're about water stress affecting plant physiology. This is why water is rarely the direct limiting factor in photosynthesis and growth. It's usually indirect through these physiological effects.
Meet Iyal, your AI study companion. Transform PDFs into smart study guides, practice tests, and flashcards for UPSC, NEET, banking, and any competitive exam.
Master your exam preparation with Iyal.